All right. Good morning, everyone. I'm Ray Cantave, Dr. Ray Cantave. I'm a senior project manager sitting in the Office of Pharmaceutical Quality. It's my pleasure to be here with you today. Thank you for having me. My talk will cover nitrosamine guidance, which is a topic that seems to come up quite often nowadays. So let's get started with some learning objectives. What do I hope to accomplish today in this presentation? Today, I aim to clarify differences between two structural classes of nitrosamine impurities, discuss the origin and the purpose of the carcinogenic potency categorization approach, or CPCA, introduced in September of 2023, outline the elements of the three-step mitigation strategy, highlight key updates from the 2024 nitrosamine guidance, and if time permits, I hope to touch on a few other ideas as well. Now for a brief history or historical background. In June of 2018, FDA was alerted to an impurity issue impacting valsartan products that led to voluntary recalls in industry, and within CDER, the formation of a rapid response incident management group tasked to take, in part, quick action to identify the root cause of this issue. 8 years later, I can tell you that rapid and quickly can be quite relative. However, our efforts were not in vain. With the discovery of NDMA and valsartan products, global regulatory agencies and pharmaceutical managers worldwide sprang into action. FDA initiated collaboration with international regulatory partners, seeking alignment on acceptable intake limits, or AIs, and testing methodologies when possible. We also shared best practices and assessment strategies through international working groups, and later participated in global initiatives for nitrosamine control, such as Health and Environmental Science Institute, or the HESI effort, which is going on today. This is because the impurity issue is not isolated to NDMA and valsartan. Within a few months, we saw the scope expand rapidly to include NDEA and valsartan, NMBA and losartan, NDMA and ranitidine and metformin, CPNP and rifampin, the gift that seems to keep on giving. Today, our investigations continue, mostly focused on NDSRIs in various products. With that introduction, let's take a step back for some background. I hope this is helpful to orient all in the audience, knowing that some of you may be more familiar than others. Fortunately for you, organic chemistry is not my strong subject, so I can keep this very, very high level. So, what are nitrosamines? Simply put, they are chemical compounds where a nitrosyl group is bonded to a susceptible amine. Since the 1970s, research has shown that these compounds are genotoxic and probable human carcinogens. Though nitrosamines can be naturally occurring in the environment and can be found in foods, they can also be synthetically manufactured in pharmaceuticals and also in rubber. So on to the structural classes. Initially, we identified small molecule nitrosamines like N-nitrosodimethylamine, or NDMA, which are primarily formed as process impurities from secondary reactions in drug substance synthesis. We will find this chemical structure of NDMA here on the left side. We have called NDMA and impurities like it small molecule nitrosamines because of their relatively low molecular weight. Later, we discovered a different type of nitrosamine drug substance related impurities, which occur primarily in drug product manufacturing when nitrosating agents, often coming from nitrite-rich excipients, react with susceptible amines within the drug substance molecule, or the API molecule itself, leading to formation of nitrosamine of a relatively higher molecular weight. You will find an example of that on the right, where a nitrosyl group, which is circled, bonded to the chemical structure of phenylephrine. So that's the API structure itself. So with a surge of voluntary recalls and new nitrosamine signals, there was an increasing public interest in the impact of nitrosamines that they might have on the nation's drug supply. This prompted FDA to communicate our understandings and recommendations through guidance documents. Our lab in St. Louis played a crucial role in our effort to respond to the nitrosamine issue as they worked through challenges in development of analytical methods used to test material like drug product or drug substance. We gained insights that shaped our guidance. As our chemistry assessors worked through issues when evaluating method validation reports, we observed sensitivity, specificity, and other method-related deficiencies. When evaluating root cause investigations submitted, we observed risks associated with raw materials, degradation reaction pathways, manufacturing conditions, recycled solvents, and even packaging. This is a big kudos to the industry for doing the research and providing us as an agency with this information that was critical to our guidance. We believe that sharing those insights will be helpful to manufacturers in their testing and mitigation efforts. As new nitrosamines were discovered, our toxicologists found it increasingly challenging to derive corresponding acceptable intake limits, or AIs, due to the lack of available, robust, compound-specific carcinogenicity data for these impurities. So importantly, we wanted to ensure that we could communicate our current understandings on AIs for nitrosamines, as this was essential for setting targets when manufacturers were doing their sample testing. To our colleagues in industry, nitrosamine guidance would communicate FDA's expectations and provide clarity and direction, which was lacking early on. There is an additional benefit within FDA, as guidance tends to enable a more consistent and comprehensive assessment. In the end, guidance development to achieve these goals is an essential piece of working collaboratively with stakeholders to address this true public health concern. So we'll go on to the evolution of FDA guidances. Hopefully you all can see this slide. We went on to publish our first nitrosamine guidance, Control of Nitrosamine Impurities in Human Drugs, or what I'll refer to as nitrosamine guidance for the remainder of my presentation, in 2020, and subsequently updated that guidance in 2021 to update a deadline. At the time, our comprehensive understanding addressed small molecule nitrosamines and described root causes and provided API-focused recommendations or drug substance-focused recommendations. Though the guidance was destined to become outdated with the quickly expanding scope of understanding, it introduced the three-step mitigation strategy, which was adopted internationally and remains alive and well today. This high-level concept provided a sequential framework for mitigation that we continue to build on today. Step 1 recommends manufacturers perform a comprehensive risk assessment to evaluate potential nitrosamine impurities in the drug product. Records of this risk assessment should be maintained on site and available upon request. Step 2 recommends that if a risk, or any risk at all, is identified, that manufacturers perform testing to confirm the presence and levels of nitrosamines. Step 3 recommends that based on those testing results, manufacturers make and report changes to prevent or reduce excessive nitrosamines per applicable requirements. These recommendations apply to chemically synthesized APIs in application products, regardless of where it is in the application lifecycle, as well as OTC products. All right, in 2023, we published the second guidance, titled Recommended Acceptable Intake Limits for Nitrosamine Drug Substance-Related Impurities, or what I'll call RAIL, the RAIL guidance, moving forward. At this time, we were increasingly concerned of the risk that NDSRIs pose to the nation's drug supply. That concern persists today. Within this guidance, the RAIL guidance, we introduced a new concept to predict AI limits for NDSRIs based on their structural features using a model called the Carcinogenic Potency Categorization Approach, or CPCA. I mentioned earlier the challenges our toxicologists faced when deriving acceptable intake limits. So this predictive model, which was eloquently described in the REL guidance, is the basis for more than 300 potential or hypothetical nitrosamines that are listed on our table 1 of the CDER nitrosamine impurity AI limit webpage. This achievement was welcomed by industry counterparts, as it provides clarity on what limits manufacturers should target. Finally, last year, we published the second revision of our 2020 nitrosamine guidance, which provides some key updates impacting NDSRIs that I'll address soon. But first, we've reached our first challenge question. All right, so FDA's recommended 3-step mitigation strategy includes all of the following except: perform risk assessment, submission of risk assessment to FDA, perform confirmatory testing, report changes to FDA as appropriate. Hands for A? B? All right, I see many hands. C or D? 100%. Thank you all. The answer is B, of course. Remember, the 3-step mitigation strategy recommends maintaining a record of your risk assessment at your manufacturing facility and available upon request, not necessarily submitting it to FDA. Now for some key updates of our nitrosamine guidance, updated last year. First, key update simply broadens the scope of the nitrosamine guidance to include NDSRIs alongside small molecule nitrosamines. Manufacturers should assess the risk of NDSRI formation, which occurs primarily in drug product manufacturing, in addition to evaluating the risk of small molecule nitrosamines, which primarily form an API synthesis, following the same 3-step mitigation strategy. Second, the second key update is related to nitrosamine controls, and you'll see it on the screen here. This update introduces a flexible approach for calculating AI limits when multiple nitrosamines may be present. Where we previously recommended that nitrosamines, when multiple are present, be controlled to the limit of the most potent nitrosamine, we are now suggesting that manufacturers instead ensure that the levels of nitrosamines, when totaled, should result in an exposure level that does not exceed the acceptable increased cancer risk of 1 in 100,000. And there's an equation on the screen there on the left side for you. Here you'll see revised nitrosamine guidance updates control recommendations with a clarification on method sensitivity to align more closely with recommendations of ICH Q2R1. When our first version of the nitrosamine guidance recommended a level of quantitation of 0.03 ppm, we found that it would be more practical to revise the recommendation such that the level of detection and level of quantitation are commensurate to the level at which the impurities must be controlled. The third key update I'd like to bring to your attention today provides some recommendations for implementing changes based on confirmatory testing results. You will see, again, that our new recommendations continue to build on the 3-step mitigation strategy of risk assessment, testing, and reporting changes. Now, let's assume that NDSRIs form. We recommend that you perform confirmatory testing. And depending on those findings, we recommend that you submit changes to your application as illustrated here. If confirmatory testing finds nitrosamines exceed the corresponding FDA recommended AI limit, we recommend that you make changes to reduce or mitigate the impurity and submit those changes to the agency for review as a prior approval supplement. If confirmatory testing finds nitrosamines exceed 10% of the corresponding FDA recommended AI limit, but does not exceed the FDA recommended AI limit itself, we recommend you implement routine testing and submit those changes to the application as a CBE 30. If confirmatory testing finds nitrosamines do not exceed 10% of their corresponding FDA AI limit, we find that routine testing may not be necessary and recommend you submit those findings as an annual report. Sharing these reporting recommendations, I don't mean to exclude that there are others that are still in place. So you'll see here that these include timely reporting of observations of excessive nitrosamine levels to FDA through field alert reports in accordance with 21 CFR 314.81, and timely reporting of discontinuances and interruptions in your manufacturing by contacting CDER's drug shortage staff in accordance with Section 506C of the FD&C Act, as this allows FDA to consider as soon as possible what actions, if any, we need to avoid shortages to protect the health of patients who depend on your products. So let's take a quick detour with the second challenge question. All right. Which of the following statements is not true? FDA recommends that applicants perform confirmatory testing if a risk assessment indicates an impurity is at a low risk of formation. FDA recommends that applicants routinely control nitrosamines if testing results indicate that nitrosamines are found at less than 10% of the corresponding applicable FDA recommended AI limit. FDA recommends that applicants routinely control a nitrosamine if testing results indicate that nitrosamine is found at greater than 10%, but less than or equal to than the corresponding FDA recommended AI limit. And D, FDA recommends that if applicants find nitrosamine impurities in their product exceeding the recommended AI limits, they mitigate and report changes as a prior approval supplement. Alternatively, you may provide justification for a different limit via PAS. I'll give a second for folks to see these responses. And anybody for A? What about B? All right, I see hands for B, C, and D. All right, you're right. So pardon if this question is a little tricky, but remember, even low risk is considered risk. All right. So I'd like to briefly bring your attention to an essential resource that we developed to be your landing page for nitrosamine-related information. For comprehensive information, visit CDER's nitrosamine impurity acceptable intake webpage, which includes links to guidance documents, acceptable intake limits, interim market-wide regulatory discretions, which are only applicable to approved products in the market, and these interim limits are not supportive of a permanent control limit. We also include analytical procedures that are posted in a section entitled Emerging Scientific and Technical Issues, where we plan to periodically communicate new information, understandings, and update timelines. And also a very useful revision table that shows each time that the webpage was updated. You'll see a screenshot here of the front of that page, which also has an opportunity for you to subscribe to receive email notifications about updates, ensuring that you stay informed on the latest developments. So back to the updates. Our nitrosamine guidance was revised to recommend some of the following strategies to mitigate NDSRI formation in your drug product, as previous versions only were API focused. So the recommendations include screening for excipients, adding antioxidants, adding pH modifiers, and exploring innovative strategies. Our scientists are eager to evaluate those new approaches that you propose. As we learn more about the root causes of NDSRIs, it became apparent to our experts that the product reformulation would be a powerful tool for mitigation. Which brings me to the fourth key update on the nitrosamine guidance that emphasizes reformulation as a mitigation tool. So when you're reporting reformulations, we recommend that you submit data for at least 3 batches, accompanied with at least 3 months of stability data. If our analysis sees the potential for an upward trend for out of specification, we might request 6 months. And long-term data for the first 3 production batches should be reported through the proposed shelf life and annual report. So in addition to these CMC recommendations, the guidance includes recommendations for alternative BE approaches that may apply to your proposed reformulation application. So with reformulation on the table, we realized that in certain cases, this type of change necessitates in vivo bioequivalent studies. So FDA scientists went on to research the impact of adding small amounts of antioxidants on nitrosamine formation and permeability. And additionally, research funded by FDA through the CERC initiative has shown small amounts of antioxidants do not affect intestinal transporter activities. All these together, as long as it's in accordance with 21 CFR 320.24, may provide alternative approaches to show bioequivalence without in vivo studies. And if you have more questions about that, you can reach out to an expert in bioequivalence or read our guidance for more details. So I have some future considerations for your thoughts. We don't know what's going to happen in the future, but these points may be important. As soon as I feel like we had a hold of the small molecule nitrosamines and the SRIs popped up, so we want to know, we're not sure what's next. So continued advancements in analytical procedures and research health impacts will enhance our understanding and support control measures. Industry partners have been doing a lot of this already, but it's important to consider these points to implement comprehensive risk assessments, especially for products at high risk of potential potent nitrosamine formation. Engage proactively with regulators and invest in research technology for effective nitrosamine control, because we're not yet out of the woods. Until then, we expect manufacturers to adopt the best practices and consider our guidance recommendations, continue to report issues as they arise and remain responsive to our inquiries, maintain your commitment to patient safety and quality control, and collaborate with us on nitrosamine control measures. So this morning, we covered the details that you see here in these 7 slides. And in closing, let's continue our commitment to patient safety and quality control by adopting best practices, staying informed through resources like the CDER Nitrosamine Impurity Acceptable Intake webpage, and collaborating to address nitrosamine challenges in the future. I extend my gratitude to the colleagues across the agency who support this work, with special thanks to Susan Zuck and Dr. Dongmei Liu for their assistance in preparing this presentation. Thank you also to the GDF committee, and thank you. If you have any questions, feel free to get in touch. Thank you. Oh, I'll pass it on to the next speaker, Dr. Kaur.